about a presentation titled "A Cloud-Based Web Application for Computational Imaging Algorithms Inspired by Optical Physics." And this is a presentation that was originally given at SPIE Photonics West under the AI and Optical Data Sciences seminar. And this follows work that we've done in Jalali-Lab at UCLA. So basic outline of the presentation is as follows. We're going to go over motivation for these algorithms and some background for our lab, and then go into the algorithms themselves, and two that we'll speak in detail about, entitled Phase Stretch Transform, and then one called Phase Stretch Adaptive Gradient Field Extractor. And then we'll go over the application structure, this web application that we're speaking about, and then go over a use case that involves using this application for live cell tracking. It's an algorithm that we've built called Cytolive. And then lastly, go over some remarks to conclude. So the basic motivation is as follows. The background of our lab is in hardware, specifically photonic and optical hardware. But we've taken this journey into the world of algorithms with the development of image processing techniques that simulate physical optical systems. Specifically, they simulate propagation of a EM signal through a diffractive medium according to the nonlinear Schrodinger equation. The claim that I'll make, and this is something I've come to find in my research, is that this approach of designing algorithms according to physical systems is really a breeding ground for some creativity and elegance of design that's difficult to achieve artificially. Besides these benefits, there remains the potential that these algorithms can be implemented in physical optical analog systems for speed and increases and power decreases. See here on the right now an example in which two algorithms are used in a hybrid form to create a feature map of the original difficult to see image. And this is a theme that we'll touch back on a little bit later. So the idea of these physics-inspired algorithms follows from the nonlinear Schrodinger equation. Here's just a simple diagram showing the basic physical features of these algorithms. Essentially, we have our incoming signal witnessing a modification by propagation through a medium with some engineered 3D dispersive property. And this is instituted by applying a phase kernel to the frequency domain of the image, which incurs some phase profile. And this can lead to all sort of filters, but we show three of which that we'll explore here in detail. And they can be seen on the right, operating on the same image. We have PST and PAGE, which are both feature extraction algorithms, and then GST, which is an image enhancement algorithm, which we sort into PST. And you can see here coming from optical physics to computational imaging. So we have our input signal here in the continuous domain, propagating through some distance z, and then that can actually be encoded in discrete domain with this PST operator on the bottom. So we actually are measuring the phase at the output, and the amount of phase applied to the image is frequency dependent and is encoded within the phase kernel, with higher amounts of phase applied to higher frequency features of the image. And since sharp transitions such as edges and corners contain higher frequency contents, PST emphasizes this edge information. And these features can be further enhanced by applying thresholding and morphological operations. So then we really have PST as this physics-based image processing approach. And in the above equations, S is the stretch operator, and then E of X, Y is the input image, X, Y is spatial frequency variables, and then K is this warped phase kernel, which is implemented in the frequency domain. L is the localization kernel, which helps with denoising, and then PST is this operator in which we're actually grabbing the angular phase out of this processed signal. And so PST has had some traction on both GitHub and MATLAB file exchange. It's been forked and used a lot. And we see here just some example of PST working pretty well on an image with kind of low light conditions, kind of impaired images, and able to pick up a lot of features and details, which is very helpful for things like object detection, etc. So these applications have been seen also in further literature after. So people have used PST for applications like digital retinopathy. It's called a local flow phase stretch transform, so it takes into account directionality. And then also object detection in the case of, for example, Chinese white dolphin classification with PST as a pre-processing. Phase stretch adaptive gradient field extractor, or PAGE, is an algorithm that follows a similar insight, but to a different end. So instead of one channel of edges as computed within PST, PAGE creates a set of directionally and frequency bin channels of edges. And so what that really means is that we get many channels where we filter out based on one orientation of edges. So this gives us the ability to pick out structural details from an image in a way that PST really can't. So it follows a similar math and similar physics. The kernels used filter out, again, the directionality of these edges. And what we see here on the left is an animation that goes channel by channel through the PAGE output. While on the right, we can see PAGE output embedded in RG space with color representing directionality. And so one thing that we've noticed with these is that these algorithms can be used in conjunction with each other for more powerful actual composite techniques. And so this leads us to the idea of using them as modular pieces in a larger pipeline. So for this would kind of correspond physically to propagating through two subsequent mediums with different optical properties. And we'd see that there is, in fact, a very large distinction between the top and bottom pipeline, the first being just running PST on an image in low light conditions, whereas the bottom corresponds to running first with some type of image enhancement in the form of GST, and then running feature extraction on this. We get much denser edge content. So the next thing we want to take a look at is a demo of this web application we've been building. So here it is actually something you can find online, and you can upload images to these libraries and kind of keep them here. So the idea is that for larger scale projects, you can actually get all of your data on here to get rid of some of the memory burden. This is stored on AWS S3. As far as the algorithms, we actually can run PST and PAGE on these algorithms, and so we can change these parameters and then actually process them. And so right now this is done locally, so it takes a bit of time, but the goal is to actually deploy these on larger GPUs on AWS EC2, and then these would be obviously a lot faster in those conditions. So we see here just PST output of this image. We can download that to our local machine or save it to our library online. So one of the use cases that we have been considering is this image processing pipeline for live cell tracking. And so this is an algorithm that we've actually used these sub-algorithms for within our lab. So we have incoming video data, which is just these frames of cells moving over time. And we run GST on them in order to enhance them. These are taken under low light conditions in order to not actually kill the cells from phototoxicity. So using this, we can actually get into a domain in which feature extraction is a little bit more effective. So we use GST to do this enhancement, and then PST to read out the features. And then with these features, we can actually do cell analytics and do cell lineage generation, things like that, which can be really helpful for applications like drug discovery. And so this is just an example of this idea of live cell tracking with Cytolive, which is this algorithm. So on the bottom, we can see the input image, which is difficult to get any features out of, and then PST output edges essentially here. And then as we're going along, we can actually see these cell lineages being generated, daughter cells coming from parent cells, and cells dying. And so this obviously can be super helpful, but these experiments take a long time actually to complete. And manual annotation is kind of out of the question just because of how much data there is. SoapProfile, which is an open source software, can do this, but it takes quite a bit of time, and you kind of have to run the whole experiment before you can do this. So the goal would be to come up with some pipeline that you can actually calculate these metrics and these analytics in real time during your experiment, so you can do early stopping, use your time and data faster and better. And so we've kind of come up with this idea of this hardware to cloud pipeline where we have some imaging instrument here. It could be something simple or more complicated like time stretch microscope, which we've developed with some type of edge device with some digital camera and cheap computer board like the Jetson Nano, which comes with a GPU. Is good for vision tasks, and then we can actually upload that onto the cloud, store all this data in an S3 bucket, and then do analytics on an EC2 instance, and then pass that back for visualization and trying to get real analytics. And so we hope that this can be, built leveraging some of the work we've already done with this application, and actually bring some impact and help to different application communities. So in summary, we've created this modular image processing tool with kind of tooled with our physics-inspired algorithms. Hopefully, these algorithms can be, built together in composite hybrids routines that help out general applications. And with the power of cloud processing and cloud storage, hopefully we'll get rid of some deterrents for doing this type of research, and really hope to build this thing out. So if you're interested, please find further updates on our lab website here. And thank you to SPIE originally for hosting us, and everyone for listening, and lastly, my collaborators for all their help.